Good morning, students. Welcome back to the next botany class. How are you all? I hope all are fine. Okay. Have you learned that previous class portion, children? Yes. Good. Good, students. So today's class, we are going to learn the next topic, children, bacteria. What is bacteria? The bacteria is the prokaryotic organism. That's a unicellular in nature. The first one, where are the regions, the presence in this bacteria means everywhere the presence that bacteria. Clear? Yes. The first one, shall I ask one question? Have you noticed the preparation of curd in your home? What happened there? What happened? The milk is changed into curd. Am I correct? What happened that one? Yes, very good. So there presence of one bacterium. Okay. So that's a bacterial name is known as Lactobacillus lactis. Lactobacillus lactis. That is a bacterium. They convert milk into curd. Am I correct? Yes, good. Why it so that? Why it so? Very good. The soreness is due to formation of the lactic acid. Okay. Anyway, we can consider the bacterium that is the friend as well as that is the foe. Foe means that's the enemies. Okay. Due to their presence of beneficial and harmful activities that are related, the bacteria is certain times that is harmful, certain times that is beneficial one. Clear? Yes. Then once again I will ask the question, what is bacteria? Bacteria is the prokaryotic organism, that's a unicellular in nature, that's a microscopic organisms. Okay? Yes. The bacteria, the first inventor, that person name is known as Anton von Leeuwenhoek. Okay? Anton von Leeuwenhoek. So this person in the year of 1676, the bacterium they discovered in the year of 1676. First, the Anton von Leeuwenhoek called the bacterium, the name is known as animalcules. Okay, the name is known as animalcules. What is mean by bacteriology? What is mean by bacteriology? What is mean by bacteriology? The study of bacteria is known as bacteriology. Clear, children? This is one of the small introduction of that one. What is bacteria? Who discovered this bacterium the first time? It's a person name is known as Anton von Leeuwenhoek. In the year of 1676, they called the name is known as animalcules. Okay. Then what is mean by bacteriology? The study of bacteria is known as bacteriology. Clear, children? Yes. So next one, we are going to learn about general characteristic features of bacteria. General characteristic features of bacteria. General characteristic features of bacteria. Okay, of bacteria. So what is the general characteristic features of bacteria means? First one, so this is a prokaryotic organism, no nuclear membrane and membrane bound organelles. Okay, that's the first point. The genetic material is called, the name is known as nuclear. The first point is, what is that first point? This is the prokaryotic organism. Prokaryotic organism. Next one is the genetic material. The genetic material is called, the name is known as genophore or incipient nucleus or nucleoid. So these are the three terms. The genetic material is called, the name is known as incipient nucleus, genophore or nucleoid. Clear, children? Say yes or no? Yes. The next one, so here the cell wall region, they present in some of these components. So that is some of the polysaccharides or mucopeptides, the molecules are the presence in the cell wall region. Okay. Then here that's the bacterial cell, they presence in two type of this mode of nutrition, children. The first one is some of this comes under in a heterotrophic mode and some of them comes under in an autotrophic mode. Okay, heterotrophic mode means there is without that is a chlorophyll content. So that is the best one example of the species is the Vibrio cholerae. Then some bacterial cell, they have, that is the bacteriochlorophyll is there. So that one comes under in an autotrophic mode. Am I correct? Yes. So that is the example of the species is the Chromatium. The example of the species is the Chromatium. Clear, children? The next general characteristic features of bacterium, the vegetative reproduction. The vegetative type of reproduction, they comes under in a binary fission and endospore formation. Binary fission and endospore formation. Clear? Binary fission means the dividing of these two cells. Okay. Then endospore formation. That's the two types takes place in a vegetative type of reproduction. Then sometimes the next general characteristic features of bacteria. In sometimes the bacteria, they shows that a variation. How will this variation takes place means due to their genetic recombinations. Okay. Through that genetic recombination, they are expressing in it. They shows that variation. Clear, children? How is the method to get this variation means through conjugation, transformation and transduction. These are the three methods of sexual reproduction in bacteria. Clear, children? Say yes or no? Yes. Good. Very good. So these are the certain general characteristic features of bacteria. Clear? Yes. Next one is shape of bacteria and flagellation of bacteria. Okay. The next one is shape of bacteria and flagellation of bacteria. First of all, what are the shapes of bacteria? What are the shapes of bacteria? What are the shapes of bacteria means? Commonly they are given that 4 shapes, children. First one is coccus, bacillus form, bacillus, coccus, bacillus, spirillum, and the 4th one is vibrio. Okay, children. So all of you see. First one is coccus form of bacteria. Second one is bacillus form. Then third one is spirilla type. And the 4th one is vibrio type. Clear? This coccus type means there also comes under subtypes. It's a diplococcus, staphylococcus, tetracoccus, streptococcus, cercidae. So these are the subtypes, they come under a coccus type. Then bacillus, this is the diplobacillus type. Then spirillum and vibrio. So these are the some of the shapes of bacteria. Clear? Yes. Next one is flagellation of bacteria. Flagellation means how many number of the flagella is present in which positions that occur. Okay. So that base they comes under in 5 types. What are the position of this? What is the type of flagella means? Monotrichous, lophotrichous, amphitrichous, peritrichous and atrichous. So if we see this one, monotrichous means only one single flagellum the presence in only one side. Okay, that's a type is called monotrichous. Next one is lophotrichous. Lophotrichous means more than 2 flagellas that is attached in any one of the side, any one of the bacterial cell. Clear? That's known as lophotrichous. The next one is amphitrachus, means more than two flagellas that is attached in both the sides. Clear? That is known as amphitrachus. Clear? The next one is peritrachus. Peritrachus means numerous flagellas that is surrounded in the cell wall surface area. Understood? That is covered by the cell surface area. That's known as peritrachus. Atrachus means without flagella. That's we are called that's a atrachus flagella. Clear, children? This is the diagram is very important for 2 mark or 3 mark question answer. Understood, children? Yes. Next one we are going to learn the ultrastructure of bacterial cell. The next one we are going to learn ultrastructure of bacterial cell. Shall I ask one question, children? What are the shapes of bacteria? Coccus. Very good. Try it, children. Bacillus, spirillum, vibrio. Okay, clear. So these are the 4 shapes of bacteria. Okay. The next one is ultrastructure of bacterial cell. Ultrastructure of bacterial cell. Bacterial cell. Clear? Yes. So ultrastructure of bacterial cell. The bacterial cell that shows that the 3 layers. Mainly that's the 3 layers, children. What are the 3 layers means? First one, so that is comes under a capsule or glycocalyx. Capsule or glycocalyx. Then second one is cell wall. And the third layer is the cytoplasm. The third layer is called cytoplasm. Are you understood, children? What are the 3 layers that shows this one? Bacterial cell. The bacterial cell shows that 3 layers: capsule, glycocalyx, cell wall and cytoplasm. Okay? The next one is learn one by one. First one, what is capsule or glycocalyx? All of you see this diagram, children. Here, the first one, this is the outermost area. Okay, so this is the area. The first one, what does mean by capsule or glycocalyx? So here, this layer, the bacterial cell, so this is the gelatinous substances present, children. This gelatinous substance, the 2 components is there. One is polysaccharides and another one is mucosaccharides. Okay. Polysaccharides, its 2 components present in this capsule or it's a glycocalyx, that is a gelatinous sheet. Okay. This gelatinous sheet, they present, these are the 2 chemical substances. Sometimes that's a 2 chemical substances are present in both. Clear? Yes. Next one, what do you mean by capsule or glycocalyx? The capsule or glycocalyx means the one thick layer. Okay, so this is the one of the thick layer of the glycocalyx that is tightly present to the cell wall region. The tightly bounded to the cell wall region, that is known as capsule. Understood? The thick layer of glycocalyx that is tightly bound to the cell wall region. Tightly bound to the cell wall region. That is known as capsule. Clear, children? What is the function of this glycocalyx or capsule means? First one. So this one is give the protections. Clear? The next one. So this is the sticky like nature. One gelatinous substance is there. Am I correct? So this substance, they attaching to the surfaces. This is the sticky nature of the cell. They attach to the substrate. The substrate lives, that is the plant root surfaces and the human teeth surfaces, some tissues region. So this is the attaching purpose that one is used. Understood, children? Say yes or no? And also to retain the nutrient medium of the bacterial cell. The retaining of the nutrient of the bacterial cell. So these are the certain functions of capsule or glycocalyx. Understood children? Say yes or no? Yes. The next one, the second layer is the cell wall region. Understood? The next layer is the cell wall regions. So here, the cell wall region, so that is the granular and rigid-like structures. The next one is the cell wall region. Cell wall. The second layer is cell wall region. Okay. The cell wall region. The cell wall is a granular and rigid in nature. Okay. The first one, what is the major function of this cell wall means? What is the major function of cell wall? So this one is providing the protection and give that definite shapes to the cell. So this is the cell wall region. The yellow color representing the area. So this is cell wall. So they give providing this protection and also they give that definite shapes to the cell. Am I correct? Yes. Next one, what are the cell wall chemical substances or presence means? So here they presence in a mucopeptides and polypeptides. Okay, what are the items or presence children? Very good. So they presence in a mucopeptides and polypeptides. Okay. Then what are the mucopeptides component or presence means? N-acetylmuramic acid and glutamic acid. So some of this 4 or 5 amino acids they together they presence in a polypeptide chain. Here 4 or 5 amino acids together to make that polypeptide chain. So these are the items or the presence in that is chemical, the component is presence in a cell wall region. Clear children? Say yes or no? Yes. Next one is plasma membrane. Plasma membrane. Plasma membrane. Plasma membrane. So here the plasma membrane is the one of this, the third layer is plasma membrane. So here the plasma membrane is made up of lipoprotein molecules. Okay, the plasma membrane is made up of lipoprotein molecules. The plasma membrane only, so this can, through this is the way only, entering and exiting of this small molecules and ions. Okay. But the entering and exiting, what is the function of plasma membrane means? You have to be right. It's the entering and exiting of small molecules and ions are through this way only going on. Understood children? Then plasma membrane, here they present in many enzymes. Many enzymes means, so here this is the oxidation of metabolic enzyme. The oxidation of metabolic enzyme means, so their presence in that is the some of the respiratory chain enzyme, and also their presence in a photosynthesis process. The photosystem, they are involving certain enzymes. That are the enzymes or the presence in a plasma membrane. This third layer. Clear? This is the blue color is the capsule or glycocalyx. Second layer is the cell wall. Third layer is the plasma membrane. Understood children? Then the once again, revise this one. Plasma membrane is made up of lipoprotein molecules. So what is the function of plasma membrane? Entering and exiting of this small molecules and ions. Okay. Then what is the other item is presence means many enzymes are the presence in the plasma membrane layer. Clear children? Say yes or no? Yes. Next one. The next third layer of this bacterial cell, cytoplasm. So the presence in a cytoplasm. The third layer is the cytoplasm. The cytoplasm is the thick and semi-transparent in nature. Okay, the cytoplasm, the presence in a thick and semi-transparent in nature. This is the region. Okay, thick and semi-transparent. So here the presence in a some of the granules, ribosomes, and some of the cell inclusions. Okay. So what are the items or presence means? So commonly write. So that's the cytoplasm inside the cytoplasmic cell inclusion and also the presence in the ribosomes. The cell inclusion means so that's a certain granules like sulfur granules, beta hydroxy, granules. So these are the granules and also their presence in a gas vesicles. Clear children? So that's also presence in a gas vesicle. So these are the items or their presence in a cytoplasm. The next we are moving in that inside the cytoplasm many items are there. That one is learn one by one. Clear children? Yes. Next one is bacterial chromosome. Bacterial chromosome. Bacterial chromosome. Clear? Bacterial chromosome. So in the center region, this is the red color representing this one. This is the bacterial chromosome. What does mean by nucleoid or genophore? So here learn, that's one of the 2 more question. What does nucleoid or genophore? Genophore. Clear? Yes. So here the bacterial chromosome is a single-stranded DNA that is tightly coiled like structures. Okay, so that's a single-stranded, tightly coiled DNA molecule. Here, this genetic material is called the name is known as nucleoid or genophore. This is the same point already we learned from that general characters. Clear children? Did you remember that? Yes, good, very good. The next one, the bacterial chromosomes. What does mean by nucleoid and genophore? This is one of the 2 more questions, children. Here, this is the ultrastructure. The example of the species is E. coli. That's a short form. The Escherichia coli. That's an expansion of this. Short form is a Escherichia coli. That's a bacterial species name. Okay. So here, one of the amazing facts is that this Escherichia coli 1 millimeter long. Okay, 1 millimeter long. When uncoiled condition means all the genetic information is stored in this length. Understood, children? Now the amazing fact, all the genetic information of this organism, the 1 millimeter long, that's when uncoiled condition. So coiled in nature, but this is uncoiled condition. This 1 millimeter long, that's fully storing in the genetic information of this organisms. Understood, children? Say yes or no? Yes. The next one, the bacterial chromosomes, the coiled in nature, that's one end is they attach to the plasma membrane. Why this attachment is in this plasma membrane region means the scientists they are believing DNA replication. After that's the DNA replication, maybe the chromosomes are they separated into 2. Clear? So that's during this DNA replication, that's a chromosomes are they separated into 2. So that's a fact only. The one end of this bacterial chromosome is attaching in this plasma membrane region. Understood, children? Say yes or no, ma? Say yes or no? Yes. Next one, learn about this. Next one, plasmid. The next one is plasmid. What is plasmid? The plasmid is the double-stranded. What is plasmid? What is plasmid? Very, very important. One of the 2 more questions, children. What is plasmid? The plasmid is the extra chromosomal, double-stranded, circular. Here, this is the item. Okay, plasmid. So this is the extra chromosomal, double-stranded, coiled, self-replicating. That's the ability to self-replicating autonomous element. Okay, so that is called plasmid. Clear, children? But what is plasmid? The plasmid is the extra chromosomal molecule. That's a double-stranded, self-replicating autonomous element is known as plasmid. Clear, children? The plasmids, number of the species to species, the varying of those numbers. Okay, so that's the numbers may be the varies are stores that 1 megabyte to 500 megabytes. So that's storing of this plasmids size. Okay, the size of the plasmids varies from 1 to 500 MB. Clear? Yes. Next one, the plasmids, they presence in a certain, that's a genes are present. Okay, the plasmids, so this like structure, the plasmids, they presence in a certain genes. What are the genes or presence means? That's a fertility genes, the resistant genes, and also some of those, the heavy metals, that are the genes are they present inside this plasmids. Clear, children? Yes. Next one, these plasmids, they produce in a toxins and bacteriocins. This toxin and bacteriocins, toxin and bacteriocins, that is produced only in this plasmid, that is not produced in the bacterial chromosomes. Clear? This is also another one type of chromosome only. Clear? So only in this regions only the bacteriocins and toxins are produced. Understood, children? Say yes or no, ma? Yes. The next one, based on this function, the plasmids are they classified into some types. Okay? Based on the functions, the plasmids are they classified into types. This is also another one. This is the one of the 2-mark question. Define plasmids. The next one is what are the classification types of plasmids? The plasmids are they comes under first one as F. So that means by fertility factor or F factor plasmid. Okay. The next one is or resistance plasmid. Okay, resistance plasmid. Then the next one is COL, Coulson plasmid. The next one is orite plasmid. Orite plasmid means root-inducing plasmid. Clear? The next one is TI, tumor-inducing plasmid. Clear, children? Tumor-inducing plasmid. What are the types of the plasmids or presence means? You have to write these 5 types. Clear, children? This is also another one 2-mark or 3-mark question answer. Understood, ma'am? Yes. Next one, we are moving, that is the next part is mesosome. Okay, the next part is mesosome. What is mesosome? What does mesosomes mean? The mesosomes are infolding projections. Okay, the infolding projections of the plasma membrane to form that vesicles or tubules or lamellae like structures. Understood, children? So the next one is the mesosome. Okay. What is mesosome? So here the mesosome means the mesosomes are infolding projections that is produced from the cells of plasma membrane. Here the plasma membrane. So to form that vesicles, tubules and lamellae. Clear, children? Then what is the function of mesosome? What is the function of mesosome means? That is helping in that binary fission and respiration. Clear, children? Binary fission and respiration. That's the major function of mesosomes. Clear? The next one, the mesosome, after that polyribosomes. Okay, the next one is polyribosomes. Polyribosomes are polysomes. Polyribosomes are polysomes. The ribosomes are the site of the protein synthesis. Clear? The ribosomes are the site of the protein synthesis. So here, the species to species, the ribosomes numbers are varying. So around let's say 10,000 to 15,000 ribosomes are they present in a species to species, the varying of this number. Clear, children? Here that's a 2 type of this ribosomes are present. One is 70S ribosomes and another one is the two subunits, the 50S type and the 30S type. Okay, so these are the type of the ribosomes who are present. The next one of this 2 mark question. What is polyribosomes? What is polyribosomes? The ribosomes are held together by, okay, the ribosomes are held together by mRNA and forms that polysomes or polyribosomes. Clear, children? So what does mean by polyribosomes or polysomes means the ribosomes are held together by mRNA, clear, and forms that polysomes or polyribosomes. Clear, children? Say yes or no. The next one, we are moving, that's the flagellum. The next one, flagellum. What does mean by flagella or what does mean by flagellum? So here, flagella or flagella. Okay. The first one, some bacterial cell. That's a motile form of the bacterial cell. Okay. Here, the hair-like projections. Okay. So this is a hair-like projection that's emerged from the cell wall region in that different various of the length. That is known as flagella or flagellum. Clear, children? Some motile type of the bacterial cell. Very good. The hair-like projections that emerge from the cell wall region. It's varying of the length. Okay. So that is called the flagellum. Clear, ma? Say yes or no? Yes. This flagellum that is measured. That is the diameter. That's the diameter is 20 to 30 μm. Okay, the diameter is about 20 to 30 μm, and length is about 50 μm. The length is about 50 μm. In that eukaryotic flagellum means, so in this question the eukaryotic, the structure of eukaryotic flagellum, you are coming to learn, that's the chapter 5. Chapter 5, there you are to learn, that's a detailed structure. Okay, just here in this place, just you are touching that eukaryotic flagellum. What is the nature of presence in that flagella? Clear? So in eukaryotic flagellum means their presence in the 9+2 arrangements of the microtubules. 9+2 arrangements of the microtubules. How will you arranging this 9+2 arrangement of the microtubules means? Just imagine this is a plasma membrane. So this is the plasma membrane. So here they arranging in a 9+2. So this 9+2, 3. So this is 4th one, 5th tubule, 6th, 7, 8 and 9. Clear? So this is the tubules. So this is A tubule, this is the B tubules, the doublet tubules, plus the center is 2. The center also 2. So this is the 9+2 microtubules arrangement. Understood, children? So this is the flagella nature is presence in a eukaryotic bacterial organisms. Understood? Each flagellum, they arises in that single fibril alone. Then next one is, what is the function of flagella? What is the function of flagella means? It's used for locomotions. Clear, children? So this is the points we write about in a write note on flagella. Next one, the last one of this part is fimbriae or pili. Okay, fimbriae or pili. What is fimbriae or pili? What is fimbriae or pili? Fimbriae or pili or pillars. Okay, pili. Yes. So here, what does mean by pili means? Ulafil is some of the gram-negative type of bacterial cells. Clear? Some of the gram-negative type of the bacterial cell, for example, enterotoxins. Yeah. So these are the bacterial cell. From the cell wall region, the numerous hair-like appendages. Hair-like appendages means outgrowth-like structures. Numerous hair-like outgrowth that is surrounded the presence around that cell surface region. That's known as villi. Clear, children? Oh, what is villi? So some of the bacteria, this mainly they comes under in a gram-negative type of bacterial cells. Clear? This gram-negative type of bacterial cell, the numerous hair-like appendages, that's mean by outgrowths, they presence around that is the cell wall surface region. Clear? That's known as villi. So in addition, this is the normal villi. Clear? So in addition, in certain cases of the bacterial cell, in addition to the normal villi, they produce in another one type of the villi. That's villi called, that's villi name is called sex villi. That's the name is called sex villi. What is the use of this sex villi means? All of you listen. What is the use of this sex villi means? So this sex villi helps in conjugation. What does mean by conjugation? The sex villi is helping in that conjugation. What does mean by conjugation means? This is one type of sexual reproduction in bacterial cell. Clear? This is the one type of sexual reproduction in bacterial cell. Understood, children? Yes. So what is flagella or what is villi? Then what is polyribosomes? What is mesosomes? Define plasmid. What are the classification of plasmid? So these all the questions are they comes under in a 2 or 3 mark questions. Understood, children? Say yes or no? Very good. So the next one we are moving in that is the milestones of bacteriology. The next one is we are moving in that is the milestones of bacteriology. In the year of 1829, C.G. Ehrenbusch, that person only, the term bacterium, this coined, this person name is known as Ehrenbusch. Okay. 1884, Christian Gram, that person introduced that Gram staining method. Gram staining method is there is very much used to separating the bacterial type, gram positive type of bacteria and gram negative type of bacteria. That method is they introduced that person name is known as Christian Gram. Understood, children? Yes. Next one, 1923, David Hyde Burgee. This person only the first edition of Bayer's Manual. Okay, the Bayer's Manual. 1928. Freddie Griffith. This person, they discovered that's a bacterial transformation. Bacterial transformation means, so in this step, so this is a transformation that comes under in one of the type of sexual reproductions. Okay? Yes. Next one, 1952, Joshua Liederberg. This person, they discovered that's a plasmid. This person only, they discovered that plasmids. Understood, children? Yes. Then next one is some one word question. Robert Koch, he is a German microbiologist, but Anton von Leeuwenhoek is the Dutch scientist. Okay, Dutch scientist. So this person is a German microbiologist. He is the founder of modern bacteriology. He identified that some of the causal organisms for that's the anthrax, cholera and tuberculosis. These are the causing that's bacteria this person is identifying. He got it that's award the Nobel Prize in medicinal field and physiology that's in the year of 1905. Clear, children? So these are the some one more question we should prepare it. Understood, ma'am? Yes. So next one we are moving there is the next heading, gram staining procedure. The next one is we are moving that gram staining procedure. The gram staining procedure, first this gram staining that's developed by that person name is known as Daniel Christian Gram. Okay, Daniel Christian gram. This shortly you write Christian gram. In the year of 1884. So here the gram-positive gram staining procedure, mainly they differentiating in that two type of the bacterial cell. One is gram-positive type and another one is the gram-negative type. How will you identify or differentiate that gram-positive type and gram-negative type? Means gram-positive bacteria. So that red, that's a very dark violet crystal is absorbed and they expressing in the dark violet colors means, so that is gram-positive bacteria. Gram-negative means the crystal violet color is not absorbed and after that counter staining time, applying the saffron, they appear, they express that, they shows that red color that microscope view. Clear children? That's the difference between gram staining bacteria of gram positive type and gram negative type. Clear? Yes. Next one, what are the steps involved in gram staining procedure? First one is prepare the smear of bacterial culture. Smear means it's a thin bacterium that is placed in the slide for that staining purpose. Clear? The next one is stain with crystal violet for 30 seconds. They adding for this crystal violet for 30 seconds. Then rinse with distilled water for 2 seconds. The next step, gram iodine for 1 minute. They apply for that iodine, so for that 1 minute. Again, rinse in distilled water. Rinse means the washing of that one. Okay, the dipping of this item into that water. Then wash in 95% of ethanol acetone. So that is, keep it in 10 to 30 seconds. Then the next step, rinse in distilled water again. The next one is applying the saffron for 30 to 60 seconds. Again, rinse in distilled water and plot. Plot means, so that's a dry absorbing material. Okay, that's for dry purpose only. Then after that, prepare that staining procedure, observe under that microscope. Clear children? These are the steps involved in gram staining procedure. This is one of the very important 3 mark or 5 mark questions. Understood children? Yes. So all of you listen carefully children. Today I will give one work. In your biobotany subject, biobotany and botany subject, you have keep one separate notebook. In that notebook, you have to draw some diagrams. Clear? What are the diagrams going to draw that note means? First one is structure of tobacco mosaic virus, and second one structure of bacteriophage. Then next one, the viruses life cycle of phage. There are 2 types. One is lytic cycle and another one is lysogenic cycle. That's 2 diagrams. Then today's portion, shapes of bacteria, flagellation of bacteria, ultrastructure of bacterial cell. The ultrastructure of bacterial cell is very important, one of the 5 mark question. It's maybe sometimes they ask that 3 mark compulsory question, the diagram alone. Understood children? If you are one time, you have to be drawing your classwork note means you will easy to draw your diagram record note. Clear children? So that's why I am telling children, please all should try to draw the diagrams in your classwork. Clear? Are you understood children? Say yes or no? Thank you students.